Good morning, my dear students. Today we will discuss about the production of somatic hybrids and hybrids and their application in crop improvement. Somatic hybrids are the hybrids produced by the fusion of protoplasts from two different plant species. The process of production of somatic hybrids is called somatic hybridization. This is different from conventional sexual hybridization method, as it involves the fusion of somatic protoplasts, not the gametes, from the hybridizing parents. Somatic hybridization via protoplast fusion has become an important tool in plant improvement, as it allows combination of somatic cells from different plant cultivars, species or genera, leading to novel genetic combination in the resulting hybrids. This technique can facilitate breeding and gene transfer between plants belonging to different species and genera by overcoming the obstacles of pre-zygotic and post-zygotic barriers normally associated with conventional sexual hybridization method. Let us take hybrid cell production. The following methods are employed for the production of somatic hybrid cells from two different plant species. First, let us take protoplast isolation. Protoplasts from two parental plant species can be isolated by employing enzymatic isolation methods. In this method, protoplasts are isolated from plant tissues by employing cell wall degrading enzymes like cellulase, hemicellulase and pectinase. Some of the commonly used enzymes are cellulase onozoc R10, cellulysin, microenzyme R10, pectolase, etc. The young mesophyll leaf tissues are most suitable for protoplast isolation, though other plant parts can also be used as source of protoplast. Sugar alcohols, sorbitol or mannitol are usually added as osmoticum to the solution to stabilize the fragile naked protoplasts and prevent them from bursting. The naked protoplasts devoid of cell wall are released gradually in enzyme mixtures, which are then washed in suitable washing medium to remove enzymes and debris. Fusion of the isolated protoplasts. The isolated protoplasts from two parents are made to fuse using different fusion methods. In induced fusion, the protoplasts are subjected to certain pressure created by fusogent. Fusogents are fusion inducing agent, which lead to fusion of protoplasts. The use of polyethyl glycol (PEG) along with high pH of 10.5 and high calcium ion of 50 millimole per liter is considered as the most efficient method of protoplast fusion, as it produces high frequency fusion product. In another approach called electrofusion method, the isolated protoplasts are fused using electric pulse. The entire operation is carried out manually in specially designed equipment called electroporator. Now let us come to the selection of hybrid cells. The population of protoplasts after fusion treatment will contain unfused protoplasts of parental species, fusion products of same species, we call it homokaryons, and hybrid products, we call it heterokaryons, formed by the fusion of protoplasts from two different parental plant species. Only the true heterokaryons are required for production of somatic hybrid plants, but these hybrids form a small proportion of protoplast mixture, usually 0.5 to 10%. So, selection of hybrid cells is important for successful production of somatic hybrid plants. The following methods are usually employed for hybrid cell selection. Selectable marker method, chlorophyll deficiency complementation, Bussell selection method, complementation method, and culturing of entire protoplasts. First, let us take up selectable marker method. Different selectable markers which confer resistance to antibiotics, herbicides, or other toxic compounds are employed for hybrid protoplast selection. Two different protoplasts possessing such dominant characters due to the presence of selectable markers, for example, antibiotic resistance and herbicide resistance, can be fused, and the fusion product can be selected based on resistance against both the antibiotic and herbicide. Next is chlorophyll deficiency complementation. In this method, two distinct homozygous recessive albino mutants are used as parents. The protoplasts from these albino mutants are isolated and made to fuse, and the fusion products are incubated for 2 months under high light conditions. Hybrid cells produce green colonies and are identified and selected to produce somatic hybrid plants. Somatic hybrid of Nicotiana tabecum and Nicotiana sylvestris has been produced by using chlorophyll deficient mutants. Next, we will take up Bussell selection method. The hybrid cells are Bussellly identified first and isolated mechanically to allow further growth in appropriate medium for production of somatic hybrid. Protoplast fusion can be carried out between morphologically distinct cells. Green protoplasts obtained from mesophyll tissues of young leaves can be fused with colorless protoplasts with distinctive starch granules. The hybrid cells containing the chloroplast along with distinct starch granules can be identified and separated from the rest of the cells. Next, we have complementation method. In this method, some properties, usually deficiencies, of the parental species are used which show complementation in hybrid cells and at the same time permit their selection. For example, the production of somatic hybrids from Petunia hybrid and Petunia parodi. The hybrid cells divide normally on MS medium supplemented with actinomycin D to produce macroscopic colonies as they acquire both the properties of parents. Next, we have culturing of entire protoplast. The entire protoplast population is cultured after fusion treatment without applying any selection pressure for selection of hybrid cells. All the types of the cells form calli, and the hybrid calli are later identified on the basis of chromosome constituents, protein and isozyme bending pattern, and DNA molecular markers characterization. Then we have hybrid cell generation and conformation. The hybrid cells after selection are induced to generate complete plants by culturing them in suitable culture medium with appropriate hormonal combinations. The hybrid plants must be at least partially fertile apart from their useful properties so that they can be incorporated into breeding program for crop improvement. The protoplast culture techniques have been refined and large number of hybrids has been successfully generated. The verification of regenerated hybrid plants is done by morphological and molecular analysis. Isozymes and DNA markers such as random amplification, polymorphic DNA, we call it RAPD in short, simple sequence repeats SSR, inter-small sequence repeats ISSR in short, and amplification fragment length polymorphism FLP often helps in confirming the somatic hybridity of the regenerated plants satisfactory. Now we will come to different forms of somatic hybrids. The interspecific or intergeneric fusion of protoplasts often produces different forms of somatic hybrids, symmetric, asymmetric hybrids, and hybrids. First, let us take up symmetric hybrids. In symmetric hybrids, the fusion product contains the complete genomes of both the parental species. The fusion of protoplasts is often performed between the distantly related and sexually incompatible plant species. The protoplast of Solanum tuberosum potato having normal tetraploidy 4 and 48 when fused with diploid protoplast of tomato with diploid number of chromosome 24 produce hybrid cells containing complete genomes of both the parents. The hybrid cells generated into pomato or topeto, which are symmetric hybrid exhibiting both the complete genomes of tomato and potato parents. Then we will come to asymmetric hybrid. The asymmetric hybrids contain a complete genome from one parent while the other parent contributes parts of its genomes. Asymmetric hybrids may occur naturally when somatic hybridization is performed between sexually incompatible parents. However, asymmetric hybrids can also be obtained even from those normal combinations, producing symmetric hybrids when genome of one of the parents is fragmented by irradiating with either ionizing radiations such as X-ray or gamma rays or non-ionizing UV rays. Cell-fertile asymmetric hybrid containing nitrate reductase gene is produced when protoplast of nitrate reductase deficient Nicotiana plumaginifolia was fused with X-ray irradiated protoplast of Atropa belladonna. The asymmetric hybrid contains full genomes of Nicotiana plumaginifolia and few chromosomes of Atropa belladonna. The nitrate reductase gene in the hybrid was contributed by Tropa belladonna. Then we'll go to hybrids. The hybrids harbor only one parental genome, but cytoplasm from both the parents. The nucleus of normal protoplasm can be inactivated by irradiating with X-rays or gamma rays. Example is the somatic hybridization between the iodoacetate treated protoplasm of Leucopersicon esculentum, we call it tomato, and X-irradiated protoplasm of Solanum tuberosum. The mitochondria of Leucopersicon esculentum is inactivated by iodoacetate treatment, while the nucleus of Solanum tuberosum is inactivated by X-ray irradiation. After fusion treatment, hybrid cells are selected which contain nucleus from Leucopersicon esculentum and cytoplasm contains from both the parents. The hybrid plants generated are male sterile containing nuclear genome from L. esculentum and choroplast from both the parents. The mitochondrial genomes are recombinant between the two parents in this hybrid. Lastly, we will have application of somatic hybrids and hybrids in crop improvement. Somatic hybridization makes genetic recombination in sexual or sterile plants possible. The protoplasm fusion has overcome the obstacles of pre-zygotic barrier during reproduction in sterile plants. The protoplasm of potato can be fused with protoplasm of Solanum brevidens to produce hybrids of practical breeding values. Genetic recombination between sexually incompatible species is made feasible with somatic hybridization technology. Interspecific somatic hybrid is produced by fusion of protoplasm between Datura anoxia and Datura discolor. This hybrid does not exist in nature, as conventional sexual hybridization technique cannot be employed, as two Datura species exhibit sexual incompatibility. Somatic hybridization can be effectively used for the transfer of disease-resistant characters from one species to another. Many disease-resistant genes like potato leaf roll virus, leaf blight, etc. have been transferred to Solanum tuberosum from the normal species. Reduction of heterosis in somatic hybrid can also be observed when protoplasm fusion is made between interspecific and intergeneric plant species. The heterosis of alkaloid content was observed in somatic hybrid of Datura anoxia and Datura discolor, and also between Datura anoxia and Datura stramonium. There was 20 to 25% increase in alkaloid content of hybrid as compared to the alkaloid present in each of the parent. Cytoplasmic transfer can be completed easily in 1 year using somatic hybridization technique, but backcrossing to effect cytoplasmic transfer may take at least 5 to 6 years. Hybridization has been successfully used to transfer cytoplasmic male sterility in many crops like rice, cabbage, tomato, tobacco, etc. Let us come to conclusion. Somatic hybridization is a powerful tool for producing useful interspecific and intergeneric hybrids, which are otherwise impossible to generate using conventional hybridization techniques. It bypasses the normal prezygotic barriers usually encountered during sexual reproduction by allowing direct fusion of isolated protoplasm from the respective parents. Somatic hybrids can be identified as symmetric, asymmetric hybrids, and cybrids. Symmetric hybrids consist of full somatic complements of both the fusion parents. Asymmetric hybrids, on the other hand, contains whole genome of only one parent, but few chromosomes of another parent. This allows the integration of few desirable genes from one fusion partner into another. Cybrids are cytoplasmic hybrids with nucleus from only one parent. The somatic hybridization, in spite of certain limitations, provide an excellent opportunity for crop improvement by allowing the transfer of important characters from one species to another, irrespective of species and genera differences.